This video is a continuation of the structure of prokaryotic cells. In the last video, I left off with the structure of the cell membrane, which is very similar regardless of what type of living organism we are referring to. There are a few fundamental differences, but by and large, um, we have phospholipids, we have um, proteins, we have carbohydrates that are going to be on the surface here. Okay. Now, the arrangement of all of these different structures, the abundance of them will vary greatly depending on the different type of cell or the different cell types that we look at. But overall, this is a good representation of what a cell membrane is going to look like. Now, when we look at the importance of the plasma membrane, it has a different function than the cell wall. So the cell wall was to help preserve the cell when we had changes in osmotic pressure. For the plasma membrane, one of its most important functions is to allow the transport of molecules and water in and out of the cells. So we have different types of movement that occur. We have simple diffusion, and in this figure here, this is simple diffusion. We have our phospholipid bilayer here, extracellular fluid up here, which would be what would be outside of the cell, and then our cytoplasm on the inside. And so what we'll see is that over time, we get to this equilibrium where the materials are diffusing. We're bringing these little particles inside the cell to reach an equilibrium. That's simple diffusion. It doesn't require any energy. It's just a passive process that occurs due to the changes in concentration outside of the cell and inside of the cell. In facilitated diffusion, we need a little helper. We need this helper protein, this protein channel that is going to help move substances in. They're still going to go down their concentration gradient. Still no energy has to be expended by the cell. Just need this little protein channel to allow that to happen. The materials can't diffuse right through those phospholipids themselves, so we just have a little tube here that allows them to travel in. Now, active transport is completely different. With active transport, we're moving substances against their concentration gradient. In other words, we're trying to move them from an area where they're in low concentration to an area of high concentration, which goes against their gradient. So we have to use energy for this to happen. So this is called active transport, where we're going to use energy in the form of ATP to move these substances against their gradient. And these forms of membrane transport, diffusion, facilitated diffusion, active transport, they happen in all types of cells. This is not just unique to prokaryotic cells. This happens in eukaryotic cells as well. Another type of movement is called group translocation. This is a method for transport into bacterial cells. It moves into the cell against its concentration gradient, but it's chemically modified so that it doesn't require specific transport. This is an example of this is what we refer to as the phosphotransferase system. It adds phosphates into or onto some material that's trying to come into the cell, and it's an energy neutral system because this is a process that has to happen anyway. Now, some prokaryotic cells, those who perform photosynthesis, have chlorophylls and bacterial chlorophyll, so pigments, that allow them to absorb light energy. And these kind of fit into the plasma membrane. Now, if we look at the structure of the cell wall a little bit more closely, so we've talked about its function, but if we look at the structure, the cell wall in bacterial cells is made up of subunits of NAG and NAM. Okay, NAG, N-acetylglucosamine, and NAM, N-acetylmuramic acid. So we call these NAG and NAM for short. And we get these repeating units of NAG and NAM, okay, that are then linked with these peptide or these protein linkages. Okay, so we have a tetrapeptide linkage here, which is 4 peptides, and then we have a pentapeptide linkage here, which is 5. And so these cross bridges make this structure pretty important, pretty tough. Okay. Now, this provides the cell wall with strength, but what you'll notice here is a very fundamental difference between what we refer to as gram-positive and gram-negative cell walls. In the gram-positive cell walls, we have these pentapeptides and tetrapeptides. In gram-negatives, we have the tetrapeptides, but then we're missing that pentapeptide link. We just have this direct link. And this is a fundamental difference between two major groups of bacterial cells, and we use the gram stain to differentiate between gram-positive and gram-negative cells. This is really the first fundamental difference between bacterial cells. And gram-positives behave one way, gram-negatives behave another way, and therefore they're treated. When we talk in terms of, antimicrobial or antibacterial agents, we treat gram-positive and gram-negative infections differently. And this is due in part to the way that the peptidoglycan, these NAG and NAM subunits, are aligned. Okay. And I'll talk about the gram staining protocol in a different video. Gram-positive bacteria have plasma membrane, just like we talked about. Gram-negatives have that. But then if we look at gram-positives, we see this huge thickness of peptidoglycan. Whereas with gram-negatives, they have a very tiny layer of peptidoglycan, and then they have this outer membrane on them. Okay. And this outer membrane contains a lipopolysaccharide, and this lipopolysaccharide contains a particular lipid called lipid A with these O antigens, and I'll talk more about these in detail. But this outer membrane can be damaging. It can behave like a toxin for some of our gram-negative bacteria. Some gram-positive bacteria also have additional structures. For instance, this is a member of the Mycobacteriaceae family that, in addition to having peptidoglycan, also have mycolic acids, which is a waxy lipid. And they stain gram-positive because they have the peptidoglycan in their cell walls, but they also stain acid fast because of these mycolic acids. And so we have a special staining procedure for them as well. So a little bit more about that lipopolysaccharide, that outer membrane on those gram-negatives. This lipopolysaccharide can function as an endotoxin. endotoxins behave in all of the same way. If we have a specific type of endotoxin, it causes the same symptoms, which would be fever, hemorrhage, and septic shock, and eventually death if we can't control that. Each of these LPS, these lipopolysaccharides, has this lipid A, this core structure, and then the O antigen. And we can use that O antigen or O side chain to identify different species and strains of bacteria. For instance, E. coli. Escherichia coli, we hear a lot about E. coli in the news, and typically when we hear about recalls involving, meat, it's E. coli 0157H7. And 0157 there refers to the particular type of O antigen. And when we have illness that's associated with E. coli, it is this E. coli 0157 that is typically at fault. So again, this is your lipopolysaccharides. You've got your lipid A here. You've got this core molecule, this core polysaccharide, and then your O antigen, and that's what makes it different among species. Now, looking a little bit more closely now at the external structure again, I've already talked about that capsule, but we can also have a different type of outer layer called a slime layer. A slime layer is only loosely attached to the cell wall, um, not like the capsule is, which is it's more organized, it's more firmly attached. The slime layer may be more easily washed off, but both of these, the capsule and the slime layer, are referred to as a glycocalyx, which is a sugar coat, and really allows the cell to be able to adhere to substances. Think about this glycocalyx as like syrup or jelly. It's this sugary, sticky substance that allows the bacterial cell to be able to attach to something in its, some type of surface in its environment, or allows it to stick to other cells. And like I mentioned, this can contribute to pathogenicity, the bacterium's ability to cause disease. Makes it harder for the immune system to identify and ingest the cell. And archaea also have glycocalyces called S layers. They're a little bit less understood. We don't really know exactly what their exact composition is and what they do. And then I talked about the fimbriae, the pili, and I mentioned, that sometimes this pilus can be used for conjugation, allowing bacterial cells to exchange DNA. And then, of course, flagella, allowing the organism to move throughout its environment.